Okay, so yeah, we're gonna first start out with, sorry, I don't know why it does that sometime. Other way. There we go. We're first gonna start out with just talking about blood typing and blood transfusions. And so let's start with this one. Okay, so this is a red blood cell, shows which antigens are there. So go ahead and just write on top on your slides there on your notes, what would each blood type be called? Okay, so it's just A, B, positive or negative. Okay, go ahead. And then there could be O as well. So write in each one. Take a second and do that with your team. And let's go ahead and keep working with our full team. So if you guys are in the front, turn around and work with the people in the back. You can be using that collective brain power that helped you so much on the quiz. Help you with this too.
I just did O negative. Yeah.
All right, let's go ahead and come back together. And instead of holding them up, I'll just call on a team with each of these questions to answer. So yeah, let's go with this question there. So the letter for the blood type that is called B negative. Who's got that answer? Who wants to raise their hand? I can call on a team too. Let's go with team 3 in the back there. Which one's B negative? Yeah. B negative would be it has the B antigen, but negative means it doesn't have the RH antigen. So team 2, want to help them out? C shows B only. That's B negative. Yeah. So we've got A negative, we've got O negative, we got B negative, we got A B negative, we got A B positive, we got A positive, we got O positive, and we got B positive. So it would be easier if it was just like an R, right? But the positive negative thing is always referring to the RH, the presence or not of the RH antigen. So positive means the RH is there, negative means it's not. A means there's the A antigen, B means there's the B antigen. Okay, if there's no antigens or negative. Okay, questions on that one? So these are little proteins, glycoproteins sticking out of our red blood cells. Okay, now antibodies are the other important part here. Okay, so now I want you to write in which antibodies would be present in blood that had this type of blood type. Okay, and these antigens on it. Okay, so talk about that as a team. Write them in there. So which is it? Anti-A, anti-B, anti-RH, and go ahead. What? No.
Because we know it's gonna be like B, but then like, would it be like B minus? Or
We call it the anti-RH antibody. So that one is a little bit trickier because we don't necessarily spontaneously produce RH antigens, or antibodies. But we typically will assume if somebody's RH negative that they do have the RH antibody because it's just safer to do the transfusion that way. That makes sense? All right, let's go ahead and come back together here. So I picked on team 2 and team 3 so far. Let's go to the back there, team 12. What do we got? Which blood type would have none of these antibodies in it? Which one? E. So which one is E? What kind of blood do you call that? AB positive, but you're correct. It is, yeah, it is that one, right? Because it has all of the antigens, right? So we would call that AB positive, and it has, it has all the antigens, but doesn't have any of the antibodies. If it had any of the antibodies, then they could stick to one of those antigens, right? And that would be problematic. So E is the answer up here, okay? Which makes AB positive blood the universal recipient, right? Because we'll talk about why that is important in a sec, but if there's no antibodies, then it doesn't matter what kind of blood we put in there because there's no antibodies for that antigen to bind to, right? So we can put any type of blood into this person's blood and it won't be a problem, okay? We'll get to the reasoning behind that next. Okay, so E was there. So with A, so it's pretty much just whatever antigens we don't see on here, the antibody is there. Okay, so anti-B, anti-RH, all the antibodies for this one, right? Anti-A, anti-RH. And now there is one caveat to that we don't typically produce antibodies against the RH antigen without being stimulated to do so. So that kind of goes into the whole pregnancy thing that we were talking about. So this person's blood here in C, they're B negative. They definitely have A antibodies. They may or may not have RH antibodies. But if we're doing a blood transfusion, we're going to assume that they do because it's just safer that way. Okay, so for all practical purposes, we're going to assume that this one has anti-A and anti-RH antibodies contained in that blood. Okay, questions on that? Yeah, Isabel? AB negative. Yeah, because they could have RH antibodies. Yeah.
like the RH antibody if you're pregnant, or can you get it?
If you're somehow exposed to the RH antigen, so somebody else's blood with the RH antigen on it, then your body will produce antibodies against that. And so, yeah, so it's typically just a precautionary thing to not give that type of a mismatch if you don't know or not. Yeah, yeah. Other questions on that? Okay, so 2 questions here. Go ahead and work through question 1, then question 2. Okay, I'll call on 2 teams to answer those.
Yeah, they are. Those are all part of
All right, let's go ahead and come back together here. Let's work our way down that column over there. So team 11 there, say Sarah's team. What do you guys think for this question number 1?
plasma also is part of the transfusion, but maybe just everything but the antibodies.
Yeah, good thoughts for sure. Yeah, and this is maybe something we don't know. Wasn't necessarily in the book. Anybody else have a guess? Yeah, it's just the red blood cells. That's what it is. Yeah, that's the primary type of transfusion is red blood cells. Then they mix it with saline solution, so it is a similar concentration to blood, but it's just the red blood cells. Okay, so that is a key thing to think about when we're deciding whether or not a blood transfusion is safe. We know that all we're giving the person is the red blood cells. So we're giving them the antigens. We're not giving them the antibodies that are in that person's blood, right? That makes a big difference, right? Because if we're giving them the antibodies, then we have to worry about all kinds of stuff, right? But if we're just giving them the antigens, because the antibodies would be in the plasma. Yeah, so that's a key thing, okay? So then what do we need to be worried about in the recipient's blood? Team 10? That you guys right here? Yeah. That's it. Antibodies. That's all we care about. We don't actually care about what antigens are on that person's blood. We need to know the antigens so we can figure out what antibodies are there. Okay. But the antigens don't have to match exactly from the donor to the recipient. Okay. So we think about what antigens we're sending to that person and make sure they don't match up with the antibodies that are in that person's blood, and then we're fine. So that's the process. Yeah, Talia? Yeah, so different types of transfusions would be those situations. So yeah, there's definitely plasma transfusions. Yeah, definitely platelet transfusions when you have yeah, a lot of times people have clotting disorders and they don't have all the right proteins. They can't make them, so then they have to get plasma from other people. So those are kind of unique types of transfusions, but a typical blood transfusion is just the red blood cells. Yeah, good question. Yeah, Ashley. Yeah, absolutely. Maybe in emergency situations, but yeah, but it is also like a very valuable type of blood too, because O negative people can only get O negative blood, right? So you don't really want to waste that type of blood. So a lot of times if there's an emergent situation, you'll just give somebody fluid to get their blood pressure back up, right? And then you can figure out the blood type and work on that a little bit later. But yeah, Alyssa. Mm, yeah, I've seen some stuff on that. And there is even stuff on like diet and blood type and stuff. I don't know if there's really a lot of strong science behind that, but I have seen some of those things too. It's an interesting question. I haven't looked into the research on it, but I'm guessing there's not a ton. But yeah, it is interesting. Okay, so let's just walk through this one together for a sec because we don't have a ton of time. Um, let's just pick a blood type. Okay, we'll do this one together. So I'll pick my blood type. Okay, I am O negative, so that means the blood bank calls me all the time. So what antigens do I have on my blood? Huh? What antigens do I have in my blood if I'm O negative? None, right? So no antigens. Okay. All right. So then antibodies present in my blood. Which ones? Could be all of them, right? Yeah, could be all. So what types of blood, what types can they give to? So what type could I give to? I give to everybody, right? Yeah, so all blood types. That was too easy of one. I shouldn't have done my own blood type. And then what types can I receive? only O negative, right? Because of that match. Let's do one more real quick. Let's do A positive blood. Okay. So which antigens are present on A positive blood? A and RH. Okay. So which antibodies are present there? Which ones? Yeah, anti-B. Okay. So which types of blood which types can they give blood to? Okay. So this is all about the antigens that they have, right? So they can give blood to anybody that doesn't have the anti A. So we'll just say it this way, can't have anti-A or anti-RH. Okay. And then which types could they receive from? So now we need to think about the antibodies present. So what's the one type of blood, or what's one type of blood that someone with A positive can't receive? Go ahead. Yeah, B positive, B negative, right? Because that's because of the antibodies present, right? Can't receive B positive or could be AB positive or AB negative, as long as that B is there, right? So the ones they can receive would be any of the O's, O negative, O positive, or any of the A's, A positive, A negative. Yeah. And so here's the deal with this. If you just like tell me your blood type and you ask me if you can receive a certain type of blood, I wouldn't be able to answer that question immediately. And so don't feel bad if you can't either. Don't think you're not getting it if you can't. Just use these steps. That's all you do. So the steps are what antigens are present on your blood. Okay. And really, we want to think about what antigens are present on the donor's blood, because that's what we care about. The antigens are going to the recipient, right? So which antigens are present on the donor's blood, and then which antibodies are there on the recipient's blood. And as long as there's not a match there, then we're fine. Okay, so, but it always takes me, I've been teaching this class for 13 years, and it always takes me having to figure it out. So that just learn those steps. There's nothing to memorize. You're not gonna memorize this table there, right? I don't think you're gonna memorize that table. Okay, just learn the steps of how to figure it out, and you got it. Okay. All right. Questions on that? Okay. Um, let's go ahead and yeah, we can do this question quickly. Go ahead. Talk about this one with your team. Decide what you think. All right, let's go ahead and come back together here. So someone with AB negative blood has a major bleed, now needs some blood. The hospital has some A negative blood available. Is this a safe transfusion? Let's go with team 7 here. What do you guys got? Kate's team? You said yes? Did the whole team say yes? Yeah. You said it's safe, okay. And explain why. What makes it safe?
AB negative would only have antibodies for RH, right?
Yeah.
And then A negative blood would have antibodies for B and RH.
Mhm.
So we said that.
Okay. Yeah. So we're not actually worried about the antibodies from this one, right? We're worried about the antigens, or we're worried about the antibodies that this person contains, right? Which is only RH, like you said. So as long as we don't have a positive type blood, then we're fine, right? Because that's the only antibody that's found here. Perfect. That's what you said too. I was just helping summarize it. Yeah. Okay, good. Yeah, any questions on that? Okay, all right, let's go ahead and do this one with pregnancy here. Go ahead and talk about this and decide what you think.
So the blood during the delivery, and then the mom feels like it's that one.
All right, let's go ahead and come back together here. Okay. Team one, I don't think I've picked on you guys yet. What's one of the right answers here? What'd you guys think? A, okay. So the blood mixes during delivery and the mom builds up antibodies. That's correct, for sure. That is correct, okay. So we don't really get blood mixing until delivery. And that's why the second pregnancy is more dangerous, right? Because the first pregnancy, yeah, we don't have the blood mixing until then, and then the baby's out, right? And then did you have another one? Trying to narrow it down. Okay. Yeah, so D would be the other correct answer here. So, oops. So during the second pregnancy, antibodies from the mom's blood can enter into the placenta. Okay. So here's the deal. Red blood cells are too big to get through the barrier between the placenta and the mom, but antibodies are not too big to do that. They can get through, right? So the red blood cells themselves don't mix until delivery, but the antibodies are just little proteins, right? And those can actually get through the barrier. So if those antibodies are already there, and then or mom has another pregnancy with Rh positive blood, then those antibodies will get through and start attacking the fetus's blood in the womb. Okay. All right. Questions on that? Go ahead, Morgan. During the delivery only, yeah. So it is the first pregnancy, but it's just the very end of it, right? Yeah. Mhm. Yeah. Okay. Question? Yeah.
So they give like antibodies.
Yeah. Mm-hmm. Yeah.
Why they give it during
Yeah, it's just to kind of to dampen the mom's response a bit. And a lot of times even on the first pregnancy, they'll do it now just to kind of be safe because they don't know what the mom's blood has encountered over the years of her life, right? And so yeah, that'll just kind of dampen the immune system's response at that point, and then again later. And there's different protocols depending on the provider as well. But yeah, throughout the pregnancy, it's given a couple times usually. So yeah, good question. Okay, so we'll run through some of these. Just we don't have much time left, so let's run through a couple of these just together here. I kind of already mentioned this one. So our first priority when someone has a big bleed is blood pressure. Okay, and so without trying to figure out the blood type, we just need to get their blood volume back up so they don't die of low blood pressure, right? So the first thing that would need to be done would be just restore their volume with something that's isoosmotic, right? Something that kind of matches the blood. Get enough fluid in there so the heart can produce a strong pump and can get blood out to the body. It might not have as many red blood cells in it. That's okay. That's better than not being able to get any red blood cells out to the body, right? So that's that one. Here we've got hemostasis, right? So the whole process, okay? So the very first step in hemostasis is what's called vascular spasm. Okay, makes sense, right? We break a blood vessel, let's squeeze it down so we don't send more blood to that broken blood vessel, right? That's the idea. Okay. Then step 2 is the platelet plug. This is like the little leaky thing that that happens. Some plasma can get through here, but not red blood cells. Then we move into phase 1. All right, phase 2, and phase 3. Okay, then we get to retraction. So clot retraction, that's taking that broken tissue and pulling it back together, bridging that gap, right? And then we have fibrinolysis. Oops, sorry, I messed that up. Fibrinolysis. That just means breaking up the clot. Okay, getting rid of it because we don't need it anymore because the tissue's healed. Okay, so those are the big steps there. Okay, one thing to definitely know for the exam is this image here, right? And all the drugs discussed here. Okay, so we can just kind of walk through the answers here because there's a lot. We don't have enough time for all of this. The correct ones here, A and C, are both related to ones that deal with the platelet plug portion of blood clotting. Okay, so aspirin blocks this right here, thromboxane A2, which is one of the things that activates our platelets and makes us form that platelet plug. ADP is also one of the things, which is just like ATP, but it's broken down a little bit, right? We've lost one of the phosphates. That also activates platelets. So the Plavix and aspirin work by blocking the platelet plug formation. Okay, so that's E is saying that as well. F is saying that as well. Same thing, stopping that platelet plug, right? And then we go down to the other ones that have to do more with clotting. So H and J, Pradaxa, known as dabigatran, directly blocks the thrombin itself, right? So thrombin does the last step of turning this fibrinogen into fibrin, and then fibrin is the main thing that makes up the clot, okay? So dabigatran or Pradaxa blocks that from doing its job, and then warfarin or Coumadin blocks the formation of thrombin by blocking one of the many steps up above. Okay, so the difference there by blocking the formation of it versus blocking the thing itself. Okay, and so yeah, those are those ones. And then we've also got K here, which is stops fibrin formation. And we've got L, stops fibrin formation as well. And then both of these are also true. Heparin is down here as well. Heparin is showing this, right? So heparin helps this antithrombin one block thrombin's job. Okay, so yeah, we can we can start off next class with this just to kind of make sure everybody's got the answers to these. So we'll do that. All right, guys, have a good rest of your day. We'll see you on Friday. All right, see you.